So good morning students. In this class we are going to discuss the blood clotting mechanism. There are two different types of blood clotting mechanism, intrinsic and extrinsic pathways. Okay, it's almost the same but it has a little difference in the beginning and remaining stages are same. So we are going to discuss it in detail. So it is very important, you have to study this, it may be asked for your examination. So, blood clotting mechanism. Coagulation or clotting, it is defined as the process in which blood loses its fluidity and becomes a jelly-like mass few minutes after it is shed out or collected in a container. That process is called blood coagulation. A clot is a mesh of thin fibril entangling the blood cells. Okay, these fibril consist of fibrin threads. The fibrin is formed from fibrin. Okay. Intrinsic pathway responds to spontaneous internal damage of the vascular endothelium, whereas extrinsic pathway becomes activated secondary to external trauma. Okay, if some external trauma occurs, then extrinsic pathway opens, and if it is internal, and spontaneous response is intrinsic pathway. Both intrinsic and extrinsic pathways meet at a shared point to continue coagulation, and they meet from a common point. Both this method of coagulation starts in different pathway and then they meet in a common point. After that they share a common pathway till the end of the clot formation. Okay, the clotting factors involved in the intrinsic pathway include 12, 11, 9 and 8. The clotting factors involved in the extrinsic pathway include 7 and 3. The common pathway includes clotting factors 10, 5, 2, 1 and 13. And you should remember the name of the each factor also. Commonly it is denoted by Roman letters, by using Roman letters, but it has some name, so you have to study the name also. First, factor 1, it is fibrinogen. Factor 2, prothrombin. Factor 3, thromboplastin. Factor 4, calcium. Factor 5, labile factor. Factor 6, which is not has been proved what is the content, and factor 7, anti-hemophilic factor. Factor 8, Christmas factor. Factor 9, stored power factor. Factor 10, stored power factor. Factor 11, plasma thromboplastin antecedent, and factor 12, Hegman factor, and factor 13, fibrin stabilizing factor. These are the factors which is involved in this clotting pathway commonly, and usually we use Roman letters, but you should study its name also. Most of the clotting factors are proteins in the form of enzymes. Normally, all the factors present in the form of inactive proenzyme. And I already shown you the factors which are involved in the clotting mechanism. All these all these enzymes which are involved in this clotting mechanism is already present in our blood, and which is present in an inactive form called proenzyme. Okay, during when this cascade opens or when the reaction starts, all these proenzyme become enzyme, that is inactive form of the enzyme become active, and which leads the reaction. Okay, this proenzyme must be activated into enzyme to enforce clot formation. It is carried out by a series of proenzyme enzyme conversion reactions. So the blood coating mechanism involved a series of proenzyme enzyme conversion reaction. So now we are discussing the different stages of blood clotting. The first stage is the prothrombin activator. Second stage is the conversion of prothrombin into thrombin, and third stage is the conversion of fibrinogen into fibrin. Only the formation of prothrombin into thrombin is different for both these pathway. All remaining stages are common for to both of this reaction, and for intrinsic as well as extrinsic. First, formation of prothrombin activator. Blood clotting commences with the formation of substance called prothrombin activator, which converts prothrombin into thrombin. which occurs through different pathways, extrinsic pathway as well as intrinsic pathway. In the extrinsic pathway, the prothrombin activator is initiated by a tissue thromboplastin which is formed from injured tissue itself. Okay, but in intrinsic pathway, in this formation of prothrombin activator is initiated by the platelets which are within the blood cells. Okay, so in intrinsic pathway, blood cells itself, from the blood itself the pathway starts, but in extrinsic pathway, from the injured tissue the pathway begins. Okay. Intrinsic pathway. Now we are going to discuss the intrinsic pathway. Okay, during the injury the blood vessel is ruptured, the endothelium is damaged, and the collagen beneath the endothelium of the damaged and the collagen beneath the endothelium is exposed. See, when a rupture occur in the tissue, the collagen which is present in the endothelium, that means the membrane of the cell, are damaged, and endothelium is damaged, and the collagen which is present below the endothelium is exposed out. Okay, when the factor 12 comes in contact with the collagen, it is converted into activated factor 12 in the presence of kallikrein and high molecular weight kinogen. Okay, so this is the first step. The collagen is exposed, then the factor 12 comes in contact with the collagen, then this inactivated factor 12 become activated 12. The activated factor 12 converts factor 11 into activated factor 11 in the presence of high molecular weight kinogen. The activated factor 11 activates factor 9 in the presence of factor 7, that is calcium. The activated factor 9 activates factor 10 in the presence of factor 8 and calcium. Then platelets comes in contact with the collagen of the damaged blood vessel. It gets activated and release phospholipid. Now the activated factor 10 reacts with the platelets on phospholipid and the factor 5 to form prothrombin activator. This needs the presence of calcium ion. Factor 5 is also activated by positive feedback effect of thrombin. And now we are looking to how this activation, prothrombin activator, is formed in the extrinsic pathway. In this pathway, prothrombin activator is formed in the tissue, and that means outside the blood. The tissue that are damaged during the injury releases factor 3, and the tissue that is the tissue thromboplastin. The thermoplastic contains proteins, phospholipid, glycoprotein, which act as a proteolytic enzyme. The glycoprotein and phospholipid component of the thromboplastin converts factor 10 into activated factor 10 in the presence of factor 7. The activated factor 10 reacts with factor 5 and phospholipid component of tissue thromboplastin to form prothrombin activator. This reaction requires the presence of calcium ions. Now, prothrombin activator is formed in intrinsic as well as extrinsic pathway. You can see how it occurs here, and you can see here. Okay. The remaining steps are common for both these pathways. Okay. So stage 2, conversion of prothrombin into thrombin. Blood clotting is all about thrombin formation. Once the thrombin is formed, it definitely leads to clot formation. Prothrombin activator that is formed in the intrinsic and extrinsic pathway convert prothrombin into thrombin in the presence of calcium. Once formed, thrombin initiate the formation of more thrombin molecule. The initially formed thrombin activates factor 5. Factor 5 in turn accelerates the formation of both extrinsic and intrinsic prothrombin activator, which converts prothrombin into thrombin. This effect of thrombin is called positive feedback effect. Stage 3, conversion of fibrinogen into fibrin. This is the final step of clot formation. and it involves the conversion of fibrinogen into fibrin by thrombin. The thrombin converts fibrinogen into activated fibrinogen. The activated fibrinogen is called a fibrin monomer. The fibrin monomer polymerizes with the other monomer molecules and form a loosely arranged strands of fibrin. Later, these loose strands are modified into dense, tight fibrin threads by fibrin stabilizing factor in the presence of calcium ions. All the fibrin threads are arranged into form a meshwork.